Please. I don't think it's supposed to be.
It's a new
All right, let's get started. Last session of the day. 2 talks to go, and the first one is Cassandra Extavour. Eggs.
Hey, thank you. Thanks to Ma and the other organizers for putting this together. Can I be heard in the back? Hear me okay? Okay. So I think I had some longer title than the program. Anyway, okay. So these are some of my favorite eggs, but I don't have access to these, so I'll talk about some other eggs.
What are they?
Oh, those are
Isn't that alien? No. Yes.
alien.
Yeah.
I wish we could study these.
Fictional.
I don't think it's.
Maybe.
I think it's aliens.
Well,
That's right. Okay.
Both
So
good
just to
movies
give
in
you
our
some context for where I'm coming from, I'm a biologist. We are increasingly trying to use mathematics in our studies, and I'll just give you an idea of some of the projects that we're doing at the moment, so you can sort of see a little bit where my current mindset is. So we're interested in process of development, how cells decide to do things. They take on different fates, they move in certain directions and certain speeds. Recently we've been moving, I guess, over the course of the last years or so from a very gene-centered approach to a more physical phenomenology approach where we're trying to quantify and characterize movements and use those behaviors to make guesses as to which types of gene networks might be involved in controlling their behaviors. So these are some of the things that we're doing. The genes themselves that control what cells do, we're interested in their functions but also in their evolution. So we've recently done a small study where there's a gene of particular interest that really, based on sequence, didn't look like any other gene that we or anybody else knew about. And so we looked at it some more and we developed a working hypothesis that it's actually a fusion of a piece of a eukaryotic domain and a piece of a bacterial domain, and they might have fused 500 million years ago and made a new type of domain combination. So we're interested in gene origin as well as gene function. We're hoping through a combination of field studies and lab studies to eventually integrate the impact of ecology and environment on these genes and their function to carry out the developmental process. And what I'll tell you about today is a project that we started recently, kind of motivated by this cell tracking project, which was done in one insect. And we began to wonder how generalizable the potential rules of behavior that we were observing in this one insect might be to the behaviors of cells in other insects. And we thought that one of the key things we were going to need to do in order to reach that goal or take a step towards it would be to characterize the eggs of insects. And so that's what I'm going to talk about today. And that will be the topic today. So size is variable. Some things are big, some things are small. Okay. Also,
I like biology like that.
right. I mean, I could say it more complicated, but it's not really better understood than that. So the particular biological things that we're very interested in are propagules, the cells or collections of cells that are the seed for the next generation. Sometimes these are what we call seeds. We use the same word. Plant propagules we often call seeds. Fungal propagules we might call spores. Animal propagules we might call eggs or sperm. They exhibit a very large size range also and a very large shape range. So this is an example of a flowering plant seed. This is the largest known bird egg, I believe, one of the flightless birds. This is sort of a regular chicken, maybe you ate it this morning. This is the longest known sperm cell in an animal. It's the sperm of a Drosophila species, a fruit fly species called Drosophila bifurca. It's hundreds of times longer, this cell, than is the body of the animal that makes it, and it's the longest sperm of any animal at all. This is the largest known plant seed. It's a giant water coconut. These are some shapes of fungal spores, shapes of pollen grains. Here's some pollen grains on the head of a bee. So these particular cells, we think it's very interesting to consider their size and shape, because we hypothesize that the size and shape of these propagule cells is going to affect the development of the organisms that develop from those propagules, because it's going to constrain the behaviors of the cells that are going to develop inside this initial structure. So that was one of the primary motivations for using this particular biological feature to study the size and shape of as opposed to something else. So the sorts of things that we are wondering is, we don't want to be measuring a bunch of bazillion eggs. Other people have done this before. How can we get that information so that we can analyze it then? There are many, many hypotheses existing in the literature having to do with physical constraints, life history constraints, evolutionary mechanisms that people have proposed to suggest that there should be rules about the sizes and shapes that are possible for propagules based on how big is the mom, or what kind of water did they drink. So we wanted to see if we could assess some of these hypotheses using larger data sets that have traditionally been used to test them. We wondered if we would discover that there might be aspects of ecology or life history or some other constraints that could predict well the evolutionary trends in sizes and shapes. We also wondered how, if we could collect a data set of eggs from organisms whose phylogenetic relationships were quite well known, if we could factor in that phylogenetic evolutionary information to inform these hypotheses. Because as you can appreciate, if one simply takes a collection of data points and scatter plots them to compare one thing with some other thing, without accounting for potentially trivial similarities among groups of those organisms that just come from common ancestry, you could be fooled into thinking that something is more significant than it is. So we wondered if we could detect any potential impact of phylogenetic correlation on some of these analyses. And ultimately, I began as a developmental biologist, developmental geneticist. I'd like to know if any of these data trends can help me make better educated guesses than I have at the moment as to the genetic regulatory mechanisms that might be underlying the evolution of these morphologies. So why should we gather new data? Because other people have asked about propagule size and shape before. Okay, there are data sets that exist. We wanted to gather them for insects, because we have worked primarily in insects as a good comparison with the fruit fly Drosophila melanogaster, a very tractable model for genetic studies. And insects are the largest clade of animals, right? There are more species of insects than there are species of every other animal type put together. But many existing data sets that have addressed propagule size and shape evolution have been based on either a fairly limited taxonomic range or a fairly recent evolutionary burst or relatively small sample size, and so we wanted to see if we could expand that a little bit. We wanted ideally a data set that would show us multiple independent evolutions of specific shapes or specific sizes, or specific other parameters, life history parameters, genome parameters, that we could use to gain statistical power to support the correlation of size and shape with some other parameter or not. If you see these things correlated multiple times independently, then there may be more support. So environmental adaptations, for example, could evolve independently multiple times in a data set that was appropriate for that. We were interested, as I said earlier, in propagule size and shape, particularly because of potential impacts on life history, development, fitness. And if we're ultimately going to get to our goal of understanding the origins of the evolutionary variation in genes that generate the evolutionary variation in shapes and sizes, we need to know what is the space of shapes and sizes we're walking around in, so we wanted to characterize this as a first step. So the people in the group who did this work are this former grad student Seth Donahue and current grad student Sam Church. They began this work as a collaboration a couple of years ago and collaborated with another grad student in the department, Bruno Guterres, and our librarian Mary Sears in the Comparative History Museum was invaluable in helping us obtain these thousands of references of published egg data that we went through. So step one is to go to the library and talk to Mary and try to get as many papers as we can where people have documented the size or the shape of an egg or both. And they would have done this in different ways. Some of them they have micrographs, some have hand drawings, some have prose descriptions. And what we did not do was read page for page all approximately 2,000 references. What Sam and Seth did, which I'll explain in a second, was to develop some custom scripts to parse through the digitized text of the literature and search for taxonomic names that we knew were insect names or insect family names, and a suggestion that there might be quantitative data about size or shape in there. And the software would extract the page of the digitized literature that it suspected contained this information, and then we manually checked those pages to see if that was correct. We chose a series of landmarks on the eggs. In some papers, these landmarks might already have been chosen, and things like length or width might already have been measured. In other cases, if there was an image, either a drawing or a micrograph with a scale bar, then we could drop these landmarks ourselves and gather the data in this way. And I'll say that I'm happy to show you the data about this later, but all the analyses that we ran subsequently, we ran them on subsamples of data, for example, to try to see the impact of different types of data reporting on the conclusions. if we excluded everything that was a hand drawing, would our conclusions change? Or were electron micrographs particularly reliable? And the answer was no. The results that we found were pretty robust to the way that the data had been reported in these papers. So some code then saved us, several thousand pages of reading, and we could hone in on the sections of the publications that might have contained data of interest, and then extract them and put them into a relational database. And these were the very basic morphometric parameters that we aimed to get. We defined length as the distance between the two longest tips, and width was the distance between the two farthest apart points on the axis that's perpendicular to the axis of rotational symmetry. Most of our, I make that specification so that you can understand that most eggs are, by this definition of length and width, longer than they are wide. But again, by this definition, there are some eggs that are wider than they are long. You think of a tomato, for example, a beefsteak tomato, not a plum tomato. It has a wider axis perpendicular to that of rotational symmetry in a second. And then we could calculate for some of them things like the degree of curvature and the degree of asymmetry, so if it was more on the pointy end or if it was more oval shaped, sort of perfectly oval shaped and so on. These are the very basic shape parameters that we began with. We're in the process of finalizing a database that makes it easy to look at and take these data if you would like to analyze them yourself. And so the idea is that here you'll be able to visualize the data. You can plot axes of these different parameters with length, curvature, asymmetry, volume, and aspect ratio as you would like. You can log transform them or not. You can choose to view them sort of color-coded by these different parameters or by these phylogenetic groupings here. And if you hover over a given point, then you'll be able to extract the image that this data point was gathered from and the original literature source that we got that data point from. So we're trying to finish this for next week, so hopefully it will be finished quite soon. And then we asked, okay, well, where are we? What is the space of shapes and sizes that we're covering, and what's the coverage of the taxonomic breadth that we have? Do we have examples from all characterized orders of insects? Insects are grouped into about 30-something groups that people call orders, and then within those orders you have families and blah, blah, blah. Okay. So the answer is yes. We have representatives from all of the major groups of insects, and this plot is just one way that you could look at these data, showing you log transform volume by aspect ratio, and these little silhouettes are sort of cartoons of how aspect ratio changes along that axis, and the different colors are different groups of insects. These colored circles, we just pulled out examples so that you can see a few things. You can see some of the ranges of shapes of insect eggs, some of the ranges of the types of data that actually exist in the papers. Some of them are micrographs, some of them, this is one of our smallest reported eggs, some of them are hand drawings, and they come from different taxonomic groups. And so we've covered all of these groups, and we've covered about 6,500 different species of insects across the clade. So let's see. So let's just start with some of the simplest things. We asked how large the eggs were and how small they were, and the smallest insect eggs are smaller than the smallest known plant seeds, and the largest insect eggs are as large as the smallest known bird eggs. These are bird eggs down here in the black dots, and then our different colors are different groups of insect eggs. And then plant seeds are over here. The biggest plant seed is going to be a coconut thread bigger than the biggest known egg. So insect eggs are not just variable in size, but they vary over a wider range of magnitude than we've seen reported before for animals. So that was interesting to us. Yes?
Is that like the biggest insect eggs?
Yeah.
Are they bigger than the insect themselves? Is that possible?
No, they're not.
They're always sort of scaled in proportion to the insect body.
They're actually not always scaled in proportion to the insect body. I don't think I brought up a slide for this, but one of the hypotheses around there is, in the literature, is that your egg, the size of the egg that you produce, scales linearly with the size of your body. We find that doesn't hold. Another one is that as you get smaller, your egg size gets proportionally larger in relation to your body size. We don't find that holds across all insects either. But no, this is the largest egg that we have here in volume. It's from a really big beetle, which is a lot larger than the egg that it makes. Yeah, and then the smallest eggs uniformly come from parasitoid insects. These are insects that lay their eggs inside the eggs of other insects. And these are the smallest ones that we've documented so far.
In the previous plot, the horizontal line is one species.
No, I'm sorry. Up here, this is just the range of documented plant seed
So
sizes.
I mean the colored lines, like the
Yeah, exactly.
Scattered.
Oh, sorry. Over here?
no.
No, I'm sorry.
So because it's the thickness of every color.
Uh-huh.
What
Is
does
this-
it represent?
Oh, the thickness is meaningless. Only the color has meaning here. Yeah, sorry not to explain that. So all the dots of one color are from the same clade of insects, from the same monophyletic group. Yeah, but the thickness, we just chose it so that it would look pretty. Yeah, yeah. The width here means nothing. Yeah.
Can I ask a quick question?
Yes, please.
It looks like the lower ends for a couple of them, not all of them, but the yellow ones and the birds
Yeah.
are kind of abrupt. I mean, there's a little bit of scatter, but is there any reason why it's a bit more abrupt on the smaller side?
Right, it's kind of, and then here's kind of trailing.
Yeah. The
That's
yellow
great.
one's quite abrupt and
Yeah, that's interesting. This group contains, mayflies and stuff like that. Here are some diptera, and the ones with the smallest ones, these are hymenoptera bees, ants, wasps. A lot of them are parasitoids. They have very tiny ones. No, I have no good explanation for that.
It does seem like with the birds, there's like just a couple that are small, but then like
Mm.
it's just a line where they start off.
Yeah, these data are from a piece of work that Maha and collaborators did quite recently, so that was a great timing that we could use this data set also for comparison and see some.
Well, just to be clear, that line means nothing because, as Cassandra said, the vertical dimension is meaningless.
Yes, that's right. I
Oh,
guess,
okay, so it's not really density. It's not okay. It's not more density, right? Okay.
The question is how representative this is of the diversity within each clade, because for birds there are dozens of species of hummingbirds, which should be on the trailing edge, not just a few. So
Right.
it depends how well each group was sampled, I suppose.
Right, yeah. I can't speak to that for the bird data set, but for the insect data set, for most clades, it's, our findings are independent of that. So if we sort of normalize to have the same amount of, or the same coverage per order, or per these, 8 or so clades that we've chosen, we find that the results are robust to that.
Yeah.
Yeah.
For example, for beetles, proportionally, there should be many more species than even hymenopteras.
Exactly right, yes. And so this is, I mean, that's a good point, you can see it right here. So the beetles are included in the orange ones, and the hymenopterans are included in the red ones. And there are more reported beetle species than any other type of insect species, but we don't find that there are more recorded measurements of beetle eggs than there are of hymenopteran eggs. And so our data set is not skewed towards the reported proportion of known species numbers.
Maybe beetles are just not as interesting.
Well, I don't know. People love bees, right? Bees have social insects, people get excited about that, so there's a lot of measurements of those things. Yeah. Okay, so this is just, a factoid. Some are really, this is the biggest one and the smallest one that we have documented. So we wanted to see, the ways that the bird egg data set has been analyzed. Some of them we can apply to ours, and some of them are different because the morphospace is slightly different. But one thing that we did want to ask were there similarities and differences between the spaces that have been documented to be occupied by these types. So this is one of the pages from one of the figures from Maha's and his group's work on the egg morphospace. There's these silhouettes of different egg types from birds, and this is sort of similar to the silhouettes of different egg shapes that we find from insects. Some of the, and these are all sort of to scale with a little millimeter mark here. Some of the egg shapes are really amazing. They have these protrusions that are, we think, not made of cellular material, but they're made of the eggshell material. It's unclear what the relevance of that is to anything, but they're there. Oh, I'm sorry about the pixelation here, but here we just tried to see, we're looking at aspect ratio on the y-axis and asymmetry on the x-axis, which is one measure of shape. And you see there's a really wide range of insect shapes outside of the bird egg space. So we don't attribute any particular significance to this, but we just observed that this is the case. The insects are, a much more diversified clade, so in many different morphological respects. So perhaps it's just one of those features as well. The type of evolutionary rate analysis that, oh yes, Ma.
Curvature, how frequent, a lot of insect eggs are completely bizarre compared to bird eggs, which are all nice and actually
Pretty
symmetric.
Yes. Yeah, so the curvature is also very, very diverse. We can have perfectly ovoid to, very, very practically half moon shape. And again, the Hymenoptera have the widest range of the curvature that we see here.
And is there a census that I'm sure you do have a census of the morphogenesis of these very bizarre shapes?
That's a great question. I can't speak directly to that, but I'll come to it in my very last slide. Yeah. Which is kind of exactly what we ultimately hope to, one of the things we hope to gain from gathering these data is information that might be informative as to how shape impacts development. I think it does. So the evolutionary rate analysis that Arhat was mentioning in his analysis of the shapes, we've done something similar here to see if there are interesting trends that we can comment on in terms of how fast do these shapes change over evolutionary time in different groups of insects. And so the yellow colors suggest that that the trees where you see the yellow colors, there was a rapid rate of evolution of, in this case we're talking about size, but you could get similar looking plots for asymmetry or curvature or volume. And I think the way that we found most useful to summarize these data are that the major clades of insects that we're considering here adopted their shape shortly after or at the appearance of their last common ancestor, and they kept that shape. It didn't change it very much once they got in. And that's reflected by some of these clades, especially these basally branching clades, the mayflies, crickets and grasshoppers, and to a lesser extent the stoneflies and the true bugs, generating one shape that was different than an ancestor that preceded them, and then holding on to that shape and not changing it very much in a dark color. But there are other groups where after diversification from a last common ancestor, like these polymetabolous insects here, those are insects that undergo metamorphosis. So they make a cocoon or a pupa or something like that. That's a derived status for insects. Ancestrally, insects we believe did not do that. And then a large and the most diverse and speciose clade of insects, including beetles and flies and bees, evolved. They evolved to have this metamorphosis, and their egg shapes are changing, have been changing rapidly since their diversification.
Cassandra, does the rate of egg size change correlate at all with the rate of DNA sequence change along these lineages?
That's a good question. There's not a strong correlation. Okay, so what about hypotheses that were in the literature to try to explain, based on some evolutionary something, the shapes of these things? So, for everything that you can think of, as often happens in evolutionary biology, any hypothesis you can think of, someone has proposed this. So it's been proposed that big eggs, as you get larger in size, you must increase in length rather than sort of staying round, so you would drop below the line of isometry, this 1-to-1 line here. And different hypotheses have been proposed, have been invoked to explain why this must be the case. Something about surface area and permeability of oxygen through an egg shell, or being able to fit inside the mom's body. Maybe if you're going to pack a lot of eggs inside, they need to get long and skinny so they can pack like sardines. They cannot be all basketballs. Okay. Also in the literature, you will find the hypothesis that no, big eggs must get wider than small eggs, depending on the size of the hole that they have to come out of the mama of, or the oviposition substrate that has been proposed to exist. Okay. And of course, you could imagine what might be a null hypothesis, which is that that you will just grow isometrically from your ancestral shape. So we asked if we considered the data, if one of these hypotheses were more strongly supported. And what's also very interesting to me, and maybe it's a legacy of some of the thinking like Dorsey Thompson and so on, is that there are some hypotheses in literature that propose that there must be very specific mathematical relationships that describe allometry of eggs and aspect ratio and so on that must hold. And we find no hard and fast ratio that is always held. And the answer to this question is any shape can be any size. Here's an example of essentially a similar curvature that scales across a couple of orders of magnitude with this data. We find this for all shapes. There are some clades that tend towards isometry. For example, beetles tend to have round eggs, whether they're very small or very large. Some groups of Hymenoptera tend to have banana-shaped eggs, whether they're very small or very large. So there are some clades that might stick closer to one of these hypotheses than the other, but overall across insects, there's no general rule here. Another hypothesis that's prevalent in the literature relates the size of the egg to how long the animal takes to develop. There is a lot of interesting evidence from different vertebrate groups that larger eggs will develop more slowly. They may have a higher hatching rate, and the idea is that perhaps there is more maternal provision of yolk, of nutrients inside those larger eggs, increasing the potential success of those offspring, but also taking longer time to use up or to fill up that space or something. It's a little bit unclear. And in fact, this is what you will get if you take the data that we had accumulated and you plotted a simple scatter plot. If you plot how long they take to hatch on the y-axis with how big the eggs are, and there is a positive correlation. The larger eggs are taking longer to develop, and we've corrected for the fact that they grow at different temperatures. So if you grow at lower temperature, you will grow more slowly typically. We've corrected for that, and it seems that you will recover this positive relationship, which has been reported many times in literature. But if you factor in how these different organisms are related to each other, understanding that, you know, if all of these dots are closely related to each other, then really I'm pseudo sampling, and I'm artificially increasing my sample size by drawing continually from the same pool that would be expected by relatedness to have the same characteristics. Basically, if we account for phylogeny, then this correlation goes away. So if we account for how these organisms are related to each other evolutionarily, there is no correlation that we can detect between the size and how long it will take to develop. So these were some of the major hypotheses that we were trying to test. First off, another thing that we tested coming directly from the bird study that recovered a possibility that flight ability might correlate with egg size in birds, and that this is correct, and that if the eggs were very large, maybe too heavy, you can't fly well. Okay, we tried to test this in insects, and there are a couple of clades of insects where wings have been lost multiple times independently based on pretty strong phylogenies, and so we could look at whether or not convergently we could detect a pattern, but we could not in the insects. There wasn't a correlation between egg size or shape and their at least ability to fly, as evidenced by did they have wings or not. So did we find any positive correlation between any trends? Here are a couple of ones that we came across.
Color over here is clade.
I'm sorry?
Color is clade.
Color is clade.
So is there some clustering of the reds and then the mauves or the purples and the blues?
That's it. Yes, that is true. I would say that's true. We haven't tried to sort of space it out, but that is true. That is true. So the Hymenoptera here have a more similar size and time relationship than the beetles do over here, maybe.
And is that functionally something that you can, or you have already ideas on? Of?
No. But you know, here the Hymenoptera eggs of similar sizes can take very different times to develop also. So, but no, I don't know what the biological meaning of this.
I mean, so
is.
Could there be a body size effect?
I'm going to
Oh,
ask.
sorry,
Yeah.
okay.
No, please.
Well, that is there a body size
That's
effect?
good. I don't know if we have looked at that. You know, we have the data, but I don't know if we've looked at that relationship.
Also, like, instead of the entire body size, the orbital size through which the eggs pass, like, maybe there is a bleeding sort of like in the microfluidic device. I don't know if, like, yeah.
Right. That's interesting. So you would want to look at that, you know, a position, duct or
that-
something with egg characteristics or with development time?
No, just the egg size
Right.
versus the tract through which it is passing when it is
Right.
actually forming that circular shape, whether that correlates. So
Yeah. So definitely, you know, I'll come in a few slides to how the eggs get made and where we might generate some hypotheses to test there. But as far as we understand, in most insects, they get made. I mean, it's not unlike a human ovary. It can grow as big as you want if it has enough eggs in it. It's not bounded by a hard membrane outside. However, when the insect egg needs to be laid, unlike in human childbirth, I mean, you would have to crack it, right? Maybe sometimes like human childbirth. So it's not an elastic thing. It's a hard, rigid, made of the same cuticle as the exoskeleton ovipositor, which can be just like a hole or like a straw through which the egg must pass. And that's definitely been something that has been suggested to constrain egg shape. We don't have a good data set for those measurements though.
In terms of life history, again,
Yeah.
sort of, larger ones hatch slower, so more yolk. Are these insects more likely to have fewer eggs? Just because more volume
Yeah, I
harder
can't
to
speak
create.
across insects. I can say for that for a small group of flies, there is an inverse relationship to the number of egg-producing units inside an ovary and the size of the eggs that are laid.
And also, therefore, the time associated with hatching, according to this?
No, the development time is not affected in that group of flies that I'm thinking of.
But over here, you do see that egg volume large also corresponds to time to hatching being large. For the pink moth, whatever that
Yes,
color
but there are also large eggs that develop more slowly.
Yeah.
Right. Yeah, and here, for example, these fairly large
Oh, I
eggs
see
can
this.
develop
Okay.
extremely quickly or extremely
Okay,
slowly.
okay.
Yeah. Arhan, what are you gonna?
I think you briefly mentioned this because I was wondering the shape and size of the egg relative to the presence or absence of the ovipositor.
Right.
Because some insects have like those parasitic wasps that lay
Right.
inside other eggs or caterpillars. The egg has to pass through a fairly
Right.
narrow tube.
A hard tube. Yeah. Yeah.
So, but I think you mentioned you don't have matching data.
We don't have
That's-
matching data.
yeah.
Yeah. One thing that I can say, at least some of the insects we work with, they have an ovipositor that is a hard tube, but it's a hard tube made of two pieces. So as the egg passes, it actually splits open like a split open straw. So I don't know how generalizable that is across straw-shaped ovipositors, to know how strong a constraint that may be or not. Okay, so here are a couple of traits that we did find that had some maybe predictability or some trends that seemed significant. So when they evolved parasitism, either ectoparasitoids or endoparasitoids, so laying your egg on the outside of an animal or injecting it inside your host, the eggs tend to get smaller and they tend to get a little longer. So here's an example of these parasitoid wasps. I said they will lay their eggs inside the egg of another insect, and then those eggs of the wasp will hatch inside the larva of the other insect and eat out the insides of the larva of the insect, come out, okay, like alien. So, and we have nice statistical power for this because shifts from non-parasitic lifestyles to endoparasitic lifestyles, that's what the purple signal here means, have happened multiple times independently back and forth. And so we find that, and again, I'm not a prime example of this, but we also have some here in the Extavour that we have evolution of parasitism accompanied by, here I'm just showing you the volume, here the size relationship, they get a little smaller, and the aspect ratio relationship, they get a little longer and skinnier. Okay, go on. And another shift that we found that there was some support for is what you're seeing in the teal here. And so this is when they shift to laying their eggs in or on water. So as you probably know, mosquitoes do this. This has evolved independently a number of different times in different groups of insects. And when that happens, their eggs tend to get a little bit smaller and a little bit rounder. And this is supported up here. These called the teal dots are the volumes for this graph here. So the sizes, they get a little bit smaller. And to some extent, they get a little bit rounder. Their aspect ratio is closer to isometry or 50 millimeters below when they evolve this aquatic aspect of their life cycle. I have no evolutionary explanations for this. I have nothing about water and sunlight and reflections and oxygen for nothing. If you guys have anything, I'm interested to hear it. This is just the observation that we made. Okay, so now that we've made some steps towards characterizing the space that we're working in, we want to move further in the direction that I'm primarily interested in, which is how genes are controlling this space, right? And so I want to tell you a little bit that we have contributed nothing to in terms of data, but this is the status of the field at the moment in terms of what might be the clues that can tell us what genes are important to determine the shape of the egg. So we have this large range of shapes and sizes of eggs, and we have to think about where the eggs get generated and when they get generated in the processes that generate them. So they are generated in the ovaries. This is a schematic of a generalized insect ovary, or reproductive system rather. There are two ovaries, bilaterally symmetrical, and there are all these other parts. These are the tubes, they have to come out of the ovary, then they have a big tube to get out of the body, and then there's some accessory business. Okay? So the eggs are made in the ovaries, which is really what we're concerned with here. And in insects, their ovaries are divided up into these chambers called ovarioles, and these are like assembly lines to make eggs. So inside each ovariole, eggs are being generated. The anterior of the ovary is at the top and the posterior is at the bottom. So the youngest eggs are at the top, and as they progress through development and become more mature eggs, they progress towards the bottom of the posterior of the ovary, and then they will go down into these tubes and eventually exit. So we want to think of the different stages of phylogenesis and how they might impact, which ones of them might impact the shape and the size of the final egg. These are just just examples from insects that we work with in our lab. This bug, this is an example of one of its ovaries of a very young bug that hasn't started to make any mature eggs yet. So there's no mature eggs in here. These are just in immature stages. They have 7 of these tubes, so varioles, to make the eggs.
What was the scale on the left?
I'm so sorry. This milkweed bug is about the size of your thumbnail. It's half a centimeter or almost a centimeter long. And this ovary takes up a third of its 300 microns, 400 microns big.
Thank you.
So if we look inside the very top part, the anterior part, it's filled with immature eggs. Each of these white dots is the nucleus of what we think is an oligonium, so an immature egg waiting to be matured. The green is a tubulin network in between all of the different bits. We don't know the function of that. And then if we move further posterior down the ovariole, we see some of these oogonia. Now the nuclei are labeled in green. Some of them will begin to accumulate. This red is a protein that's specific to germ cells, and they're accumulating this protein and developing into young oocytes. So these young oocytes here, and they're surrounded, each developing oocyte, by a layer of cells that are not germ cells, but they're important for the oocyte to develop. Morphological cells, you can find the nuclei in green. As we move even further down, here we're seeing only the tubulin in red, the outline of the cells that surround each oocyte, and then the inside of the oocyte itself is lined here in green. This is actin protein. So the stages of oogenesis are progressing from anterior at the top to posterior at the bottom. The eggs are getting bigger and bigger. And then finally, at the very end of that tube, when oogenesis is finished, that's when final shape and size will be determined. And after hatching, in most insects, the size and the shape doesn't change. And so this is what these eggs look like. And all of their little features, you can see that these eggs have a little, looks like a crown at one end. That's at the anterior end. These are holes called micropiles. These are the places where the sperm can go in. The sperm cannot just go in anywhere like in a human egg. There's holes because the eggshell is hard. So these features are all part of the shape. They're all defined during the oogenesis process. So do we know anything about genes that can change or control the size and the shape of the eggs? We know relatively little, and almost all of it is from the labs of David Builder and Sally Horn-Badovanek, Berkeley and Chicago respectively. So they did a nice screen looking for genes that, when mutated, change the shape of the eggs. And they were focused specifically on round eggs. So this is a wild type egg of Drosophila melanogaster, and it has these sort of appendages on the anterior dorsal side, but it has this normal ovoid shape, and in some of her mutants, they were extremely round. And this round shape, when is it determined? It's determined in the middle of oogenesis, when the egg is trying to form itself. And so maybe to understand genes that control egg size and shape, we need to be looking at genes that affect these pretty early embryonic processes. So these 4 panels down here, this is a wild type egg, kind of in the halfway point of oogenesis in Drosophila. And then F, G, and H are just examples of 3 different mutants in 3 different genes that she found that, when mutated, cause the egg chamber to be round instead of nice and elongated as it normally should be. Another interesting aspect that we might look to, and I don't know how far this is, we don't know yet how far any of these gene networks are conserved.
Quick
Yes,
question on the previous slide.
yes.
These are the eggs, right? There's no, like, the chamber is not being depicted, right?
Yeah, the whole assembly line is not being depicted.
Okay, so just the
Just
eggs.
one egg.
One egg, okay.
Exactly.
And
Yeah.
in contrast with bird eggs over here,
Yeah.
is the shell, well, when is the shell put together?
Yes, the shell is deposited basically at the very end of the process.
At the end, just like so there's an equivalent of a shell gland in birds, in the insects, there is a...
Not really. The shell is made of proteins that are secreted by these cells that we see here in red that outline it. Yeah, so at the very end, they're always on the outside of the egg until the very end of oogenesis. Then they secrete proteins, and then they die, leaving behind this protein coat.
And initially the egg just grows by accumulating yolk as it's moving through the ovariole.
Exactly, as it's moving through the ovariole, most of the change in size is due to yolk accumulation. And the yolk accumulation is coming in. I'm the developing egg, and out here is my fluid of the female abdomen. Through pinocytosis, through tiny gels in my cells, the yolk protein is coming in from out there, and the hemolymph coming in.
Okay. So similar to what happens in the avian egg.
Absolutely. Yeah. So there's genes that are important for this, and there's also an interesting dynamic cellular process that controls at least some aspect of the shape. So one of David Builder's collaborators in his lab discovered this a while ago. In this cartoon, we're looking at one egg in the developing assembly line. It's about the halfway point of making an egg. The germ cells are in gray, the yellow are the cells that surround it, and the green is extracellular matrix. This means a bunch of proteins that are secreted by the yellow cells and they sit on top of the yellow cells. Okay. And then this dotted line here is showing you a cross section that's illustrated here. And what this arrow means is that you can think of it, whichever way you like, the actual germ cells, the egg, the gray content inside this surrounding layer of follicle cells is rotating inside that sheath, okay? And in fact, the follicle cells themselves, the yellow cells, are rotating against the green layer of protein that they put down. And you can observe this in these amazing movies where you see these egg chambers spinning around. It's been, some of the proteins that make up the green layer have been identified. These proteins have to be arranged in specific orientated ways. You see these green lines are lined up. That's not a random little drawing. They have to be oriented in a particular way. If their orientation is disrupted, or if there's not enough of them, or if you prevent a handful of other cell movement players so that you stop these egg chambers from rotating, the egg will not elongate. It will stay round. So there's something about pressure exerted by these proteins, which are fibrous, on the outside of the egg while it is rolling. Like if you've ever made, pizza dough, cookies, plasticine, you roll out your thing and then you roll it by making it like this. This is what has to happen, at least in this insect, to make the part of the shape of the eggs. Yes?
I'm going to try to explain the question.
Yeah.
How do you distinguish, like in insects, the eggs, the polarity or the shape of the egg is defining axis polarity later on, but it's not the case for vertebrates, for example, for fish or mouse. So here, how are you distinguishing shape versus establishment of polarity?
Yeah, that's a great question. You can establish it by letting the egg develop a little bit. So these eggs here, whose shape is deformed because these mutants are wrong, they have not entirely lost polarity.
Okay.
Right? They still know which should be anterior, which should be posterior, which should be dorsal and ventral, but there are two spots. Yeah, those things turn out to be separable at a molecular level. Yeah.
So these guys have anterior, posterior, dorsal-ventral polarity. Okay.
Okay, so there are definitely some important gene regulations in the middle of oogenesis. There are some dynamic cell processes and mechanical forces that are involved, and we hope that in the future we can use this information combined with our morphospace to make educated guesses about hypotheses to test in terms of the evolution of the genetic regulation of the size and shape. I'll mention one final thing before I come to closing, which is that in different insects, there are different types of cells that probably have some impact on making the egg. I've talked about Drosophila oogenesis, which falls into the category that you see here in the middle, where the developing oocyte is the yellow cell, and the cells that surround it are these blue cells. But there are also a bunch of cells here, these purple cells, that are creating a lot of mRNAs and proteins that they're pushing into the egg. And it's important that they do that to acquire polarity and to acquire, we think, some aspects of shape and so on. But ancestrally, insects lacked this special other cell population. So every oocyte needs to make whatever it is it needs by itself. So that's this character called panellistic. And in yet other insects, like the milkweed bug that I showed you initially, each oocyte, again, doesn't have a dedicated neighboring special cell population. These are called nurse cells. To feed it a bunch of stuff. There's a shared collective pool of nurse cells up at one end that are connected to each oocyte by tubes, these purple lines. And through these tubes, they are feeding material in. So we don't want to discard the possibility that the mode of oogenesis, you have the special cells, if you have them, where are they relative to your egg? We don't want to discard the possibility that these ovarian modes may also have an impact on the evolution of shape and size of eggs. And in fact, our preliminary analysis suggests that there are some shapes that are more easily achieved with one or the other type of oocyte. Okay. So to wrap up, these questions that we were interested in, we have done a semi-automated pipeline that can help us gather a lot of data without having to manually measure insect eggs again. We're not finding support at broad phylogenetic levels for some of the long-standing hypotheses about propagule size or shape evolution. We did find evolution of specific substrates, water, other insect eggs, as accompanied by predictable changes in size and shape. Correcting for phylogenetic relatedness is very important, we found, to really evaluate carefully some hypotheses about, for example, developmental timing and size. And the genetic molecular mechanisms, of course, we don't know what those are yet, but we think we know the direction that we need to go in for this. And then ultimately, I'll just end by saying that, of course, our goal is to see whether and how the shape of these eggs and their sizes impacts how the embryos are going to develop inside them, and this is very much ongoing. But the way that we've been approaching this is to take an insect that we work with. This is a cricket embryo. It's a transgenic cricket. And so each of its nuclei is marked with a fluorescent protein, so we can watch the nuclei developing. And we know that the ways that the nuclei need to move inside the egg is almost certainly going to be different in space and time depending on the shape of the egg, because each egg starts out with just one nucleus in one place, but has to generate many nuclei to fill up the whole space. So the dynamics of that space filling may be different and in part governed by the shape of the size. We've developed a computational model that makes a small number of assumptions about the information that the nuclei must have in order to govern their behavior based on our observations. And this model explains quite well the behavior of nuclei in the one insect that we've studied, this cricket. So these are computationally generated images of the number of nuclei and their positions over time in our model compared to actual data, and I'm happy to show you more offline, other comparisons. And so we're now working on whether or not this model might be good to predict nuclear cell behavior in insect eggs that have different shapes and sizes, and now we know what types of constraints we should apply on the shapes and sizes when we plug our model into it, because we've defined that space of shape and size within insects. So these are examples of how our model thinks an egg of the shape and size of a fruit fly, or an egg the shape and size of a locust, which are very different in shape and very different in size. And let me just say, these are not to scale. The fruit fly egg, the shapes are correctly comparable, but the scales are not. The fruit fly egg is a lot smaller than the locust egg. But we have very different predictions based on the model about what the nuclei should be doing, and so now our next step is to go in and actually observe these nuclei in the real fruit flies or real locusts and see if they conform to our predictions. So the group has been great and supported the work. Thank you for your questions. I'm happy to take more, but I know we don't have a lot of time, so I'll be here tomorrow also.
I can squeeze in a question or two.
Yeah.
So the Pilgrim group, did they check if the ovariole diameter is changing or not with the gene mutations or whatever?
Overall ovariole diameter, like the ovary, like the whole chamber, is governed by what am I looking for? The combination of eggs that are inside it, like what stages are inside that chamber. The ovariole sheath, if you like, is flexible, like a stretchy shirt or something. If there are late stages of oogenesis inside it, it will be very fat, and if not, it will be very small.
Do insect eggs sort of have to act as dispersal mechanisms? You know, so I think seeds obviously all the time, and they're the elaborative things on seeds or pollen both like help them be dispersed in ways.
Yeah, there are definitely some clades of insects. Stick insects come to mind, for example. Their eggs, if you put an egg on the floor and stamped on it like this, you probably wouldn't crack it. They're like rocks. Birds eat those a lot and poop them out all over the place. And it's thought to be an important dispersal mechanism. So there are definitely some insect eggs are being dispersed.
There's no correlation between shape and dispersal mechanism?
We have not checked. We should check. Yeah.
Last one.
In chicken, the eggs also is rotating like what you showed for the.
Oh, I didn't know that.
Yeah. So I was wondering.
Is that important for the size, for the shape
For
of
the
the
shape,
egg?
yeah.
Wow, I didn't know that. But are chicken ovaries very organized? I thought they were just these kind of disorganized eggs. I don't know for sure. Most, yeah, many mammalian.
It's actually quite
many
generalized because
Yeah.
there's a region where the shell is
Yeah.
added and so
Okay.
yeah, it's not
It's
that
like an
that's
assembly
also
line as well.
the rotation controls the future orientation of the embryo.
Okay, thanks.
All right, thank you again.